to remind you that for part 6, right, that gene therapy and microarrays can be really helpful methods, and CRISPR, don't forget CRISPR. So those 3 can be really helpful methods as you start to brainstorm for part 6 of your paper, right? You need to propose 4 experiments, or 5 if you're a grad student, to try to improve either diagnosis, treatment, or understanding how your protein functions, either the wild type or the mutated form. And so that's why I wanted to kind of get these PowerPoints and these short assignments to you this week to kind of help, you make sure you understand and have those methods kind of to add to your table of techniques. So you also have your table of techniques. Those methods can also be really helpful. Maybe yeast-2 hybrid. Maybe you want to find out what protein-protein interactions occur. So places to kind of look to try to get ideas for these experiments. First off, just a reminder, you're not looking for things that are already published. These are supposed to be your own ideas. Now, I will say this question comes up every year. What if it turns out that somebody really has already done that kind of experiment? That's okay. The idea is, though, that I shouldn't find an exact replica of what's already published, right? I'm not going to be scouring the literature to see if someone's ever done it before. That's not the idea. But it just should be in your own words, really as simply put as possible, nothing fancy, but just going through why the method would be a good idea, why it would make sense, just a little bit of details about how you would set up the experiment. And that's where those 3 files that are labeled tips for designing gene therapy, CRISPR, or microarray experiments are going to be be really, helpful and useful for you to know how much detail to include, because they walk you through what to include. Obviously, this is the outline, so you don't have to actually even write full sentences for this next assignment. I just want to see what ideas you're coming up with, and then I can give you feedback as to whether or not it's gonna be like a workable idea, and if it's not, I can help you think of an alternative, right? So that when you write that final part of your paper, we at least sort of know that the ideas are going to be reasonable ones, and that you'll be able to get good points in that section of the paper. So that's the idea of the outline. But even if you have questions before the outline is due, because the outline is not due until Sunday at 5 o'clock. So if you have questions, between today and Sunday, just you can email, or you can ask me for a one-on-one Webex. If you want to talk through some ideas and ask if they're okay, that's fine. Happy to work with you. We do have some students who are still working on finishing up sections 4 and 5. That's fine too. Everybody kind of has a different situation. So, we're being pretty flexible, but we do have the end of the semester as kind of a firm end of the semester. So, we want to keep pushing along as well as we can. And I'm trying to get the feedback on sections 4 and 5 to people as soon as I can. So I'm just, I'm not waiting till I have them all graded. I'm just sending them and emailing them back with the edits as I grade them. And I'm just doing that in the order I got them. Okay, any questions, gene therapy, or if you've looked ahead to microarrays, or where to find information? Again, unmute, or you can type a question.
Okay. Someone asked if you could go over number 8 on the assignment that was due today.
You guys are very quiet today. I think the other, question 8. Oh, thanks, Miles. I didn't either see that or hear it. Okay, so let's see if I can do this without my computer crashing. So the 414, question 8. So question 8 was about methods that can be used in cases where you have a gain of function mutation as the cause of the disease. So there was a list of these under the approaches to gene therapy. And some of the methods they mentioned were RNA interference. So that one we know about. That makes sense because if there's a gain of function mutation, that often means there's going to be too much of that protein being expressed. So you would want to try to think of a method where you could actually decrease the amount of gene expression. So RNAi is good at that. And some other methods that similarly would decrease the amount of gene expression would be ribozyme gene therapy. There's something called triple helix forming oligonucleotide gene therapy. Just generally gene silencing so that there's no protein made. There's something called spliceosome mediated RNA transsplicing that was listed in that section, another way that you could decrease gene expression. And then the last one that I saw in that section was repairing the mutation in DNA by replacing the bad copy with a good copy. Right, that's gene editing, or adding in some kind of mutation so that would stop gene expression. So that sounds a lot like CRISPR to me, if you're trying to actually replace the DNA that is existing there with some new kind of DNA. That was question 8 of the 414 assignment that was due today. What I'll do is I'll post that the key to that, under the assignments. Okay. Any follow-up questions about that or any other questions? All right. Well, I'm not seeing more questions. I'll just remind you, I keep talking about those tips sheets, right, on how to design the experiments. I'll just remind you where they are. They're in the main research paper overview folder. It's the second filed down. Yeah, yeah, I agree, right? Everyone's much chattier when we're in person. I keep thinking about all the things we've lost, right, with this. And I mean, right, it's easy to dwell on the things we're missing and losing, but in terms of class, like there's this really cool hands-on microarray simulation activity. It's like a dry lab thing that I usually do. So I'm really bummed that we're not doing some of the cool hands-on stuff that we usually do at this part of the semester. And I'm definitely missing the chattiness and the questions, right? Everyone benefits from the questions. But hopefully we're making our way through. You can always also ask about any of the edits you get back on the revisions. I would suggest if you are if you get stuck as you're brainstorming for the outline, or just when you have time to start making those revisions, before we do peer editing on April 23rd. Oh yeah, and in the lab you guys are missing out on not being able to do CRISPR, right? Right. Yeah, there's a lot of things we're missing. It's a definite bummer. I don't know what else to say about the missing things. It's just a bummer, that's all. So we'll just try to get back to focusing on the positive. I think you guys are doing great with the writing and finding papers. I'm really impressed. It's not always perfect, and that's okay. I'm letting you know, like if you picked the wrong paper, or if it wasn't a cloning paper, I'm just letting you know, hey, this one doesn't quite work, and instead use this other one. So and I'm not subtracting any points or anything like that for just, for not picking the correct paper. The whole point of writing the draft and getting the revisions is a little bit working on writing, and also, just making sure that you have the right papers. Miles, do you have a question about microarrays? Or you just want to talk about microarrays? I could say a few things about them. that's probably a method you guys haven't heard about so very much, maybe in any of your classes. Yeah. I was going to mention them, so thank you. What I was going to do was pull up the PowerPoint and the assignment just to see if there was anything, um, that kind of caught your eye that looked like it was difficult. But I'm not going to pull up any more documents because that seems to shut down the WebEx on my end. But microarrays, I'm just going to tell you, are a really, really cool technique. They allow you to look at gene expression at a genome level. And so people call that like a transcriptome level, because you're looking at levels of RNA and how levels of all the RNAs in the genome change, for example, in a tumor compared to a healthy cell. So it's all about making a comparison with microarrays. You're going to say, okay, in the healthy cell, all these genes are expressed at whatever those different levels are, and they change. Some of them are going to be expressed at higher levels in the tumor, and some of them are going to be at lower levels in the tumor. And you get this huge data set about gene expression for all these different genes. So it's a really powerful technique. There is another technique that's very similar that's called RNA sequencing that can get you the same kinds of information about how levels of RNA change between two samples, like your control and your experimental. So what you can do with that kind of information, there's potentials to improve diagnosis, there's potentials to improve treatment, and there's potentials to better understand how your gene functions. So that's all spelled out in the PowerPoint, the narrated PowerPoint, which is the 414 PowerPoint. It goes along with the 416 assignment, right? So you can look at the PowerPoint today or when you have time, and then it's a 7-question assignment that's due Thursday. And again, in terms of thinking of microarray experiments that you could do, they will work for every single disease. So that's why I like to mention them and definitely kind of draw your attention to microarrays, because every single student can use this method in some way. Doesn't matter what kind of disease you're working on or what kind of protein you're working on, they can be helpful. So take a look at that this week. And if you have questions, make sure, again, you look at the tip worksheet to that kind of walks you through how to design one of those experiments. And then, see how you're doing with that and see what you come up with. And then let me know if you have questions. What is the NCBI thing there? Oh, finding expression patterns. Gotcha. Yeah, the NCBI site has a ton of information, and I'm sure you guys are probably Googling, because I would if I were you, to try to get some information. And there is a lot of information. But for part 6, make sure that you are not actually looking for too many papers at all, and rather just try to think about something that would be possible that a person could do. I don't expect that in part 6 you are going to really come up with something 100% novel, not necessarily. I don't, but I don't want you to go read about something and then write about it. I want you to kind of try to come up with it on your own. And I'll probably be able to tell depending on the way you write about it. So just don't go looking for papers for part 6. Just think about these different methods we've learned about all semester and these new ones from this week, and see what you think might be interesting to do in, like, if you were a researcher, a clinical researcher in the lab. And it doesn't even have to be like, I don't know, like if you can see some flaws in your own experiment, like, oh, well, you could do this, but I'm not sure it would 100% make sense, that might be okay too. It has to be reasonable in the sense of, like, for example, if you are working with a disease where there's a gain of function, right? There's too much protein being expressed, then proposing a gene therapy experiment where you want to add in more protein, that's not reasonable, right? That's the kind of thing we want to avoid. That's not going to make sense that you want to add in like more wild type protein when there's already too much being expressed. So that's the one kind of thing we want to avoid, where it just doesn't make logical sense. But other than that, I'm not expecting, you know, within a short, you know, week or two period of time that you're going to come up with an amazing kind of experiment to propose. Okay, so the bar is not too high. We just want to make sure that you're applying some things you've learned over the semester. Questions while we're talking about that? Does anybody have any questions about part 6? All right, I'm not seeing more questions or comments. I guess that's all I've got for today. Hang in there. We're almost there. What do we have? So today is the 14th. So 3 weeks? 3 weeks, right? A little less than 3 weeks. So you're almost there. Try to keep up some motivation. And please touch base with me if your timelines are going to be any different. Like if you just need extensions on different things, just touch base with me by email. And I hope everyone stays well. Yeah, right. I see Sam's point. He doesn't have questions, but he was hoping other people had some. I think you will all have questions potentially once you start working on part 6, which I realize you're probably just going to be getting started on that this week. So just let me know by email when those questions come up, okay? Thursday, you know, we'll be back in class like this, and maybe by Thursday you guys will have some more questions, okay? So look at those tips sheets before Thursday, and I'm going to tell people maybe to try to do that. And hopefully Webex will be a little bit more stable so we can look at those together. But go ahead and try to look at those before Thursday to see if they make sense to you, and if you understand how much detail you want to be putting into those, okay? All right, so try to come with some questions for Thursday. See if you can brainstorm at least a couple ideas on your own and see where you're getting stuck. And good luck with that. Hopefully other classes are going okay. And I will see you guys Thursday.